All right, everyone. Thank you for coming. We have some folks on Zoom, so I just want to remind the Zoomers to put yourself on mute so that we don't hear feedback in the room. I'm Ellen Hahn. I'm the director of UK CARES, Center for Appalachian Research and Environmental Sciences. Really glad you're here. We're so excited to welcome Dr. Matt Cave from right down the road at U of L. Matt's actually a graduate of our College of Medicine. I guess I can tell you when, but I won't bother with that. But he is an associate professor. It's right there, actually, in the Department of Medicine at University of Louisville. I guess he didn't wear his red. That's good. And I'm only going to emphasize the Division of Gastroenterology, Hepatology, and Nutrition. But apparently, he has a lot of other departments that he is with. He is a staff physician for the VA over there at U of L. And I just wanted to mention that did I say this already? That he is a graduate, with high distinction from UK College of Medicine. And among his many other accomplishments, I just want to emphasize that he has an R35 from the National Institute of Environmental Health Science, which is a river grant. River stands for something, maybe you can tell them what, but it's really prestigious. It's a prestigious award. It's a 7-year, 8-year project and that is very highly competitive. And so we are so excited he's here to share his wisdom about environmental health.
Well, thank you.
Yeah.
Up in the corner was the last time I was here in Lexington. So my son is number 69. So he made it up on the Wildcat scoreboard playing for Trinity High School in the state championship game. So I'm a proud father, and he's going to be attending UK in the fall, but he's not going to play football. So I don't know what I'm going to do with my, I guess, Saturdays now. I don't know what I'm going to do with my Fridays, but I think it's better for his brain. But that was the last time I was on campus about a month ago. So I like to start off talks with quotes from Wendell Berry. Wendell's a Kentuckian, lives in Henry County, environmentalist, novelist, poet. This is the Kentuckian's golden rule. Do unto those downstream as you would have those upstream do unto you. So I am a clinician. I do have some industry relationships related to clinical trials. None of those are really relevant to this talk. This is a picture of my family's farm in Shelbyville, or Shelby County anyway. My sister has horses in this barn, and there's a rainbow there, but what you can't see is the field of Roundup Ready corn immediately behind the barn. So this is exposures, or something that affect all of us, including my family. So as a clinician, I like to start off talks with a case presentation. And so this is a clinic patient that I see. And I can talk about him freely because he signed agreements that I could do so. So this case is actually published in Discover magazine. They did a feature about some of my research. So if I get published in a journal, mother doesn't care, but I made Discover magazine. That was a big time. But anyway, this was one of my clinic patients. Paul, he's a retired Marine with liver disease, steatohepatitis, and cirrhosis. He reports exposures to volatile organics, including vinyl chloride, and drinking water at U.S. Marine Corps Base Camp Lejeune in the 70s. He has risk factors for liver disease, including diabetes and obesity. And interestingly, Paul had a brother who was also my patient. He was my patient for about 2 or 3 days before he died. He came to the hospital really sick, died of liver cancer and cirrhosis. And he also had vinyl chloride exposure. He worked at Louisville's Rubbertown manufacturing complex. So 2 brothers, both had exposures to a chemical, both had liver disease. And so Paul wanted to know, what's the contribution of his exposures to his current liver disease. So objectives for the talk: I'm going to review occupational and environmental hepatology, which is relatively new and understudied field. And then talk specifically about liver disease that I call toxic and associated steatohepatitis, or TASH, and how that can be related to many types of exposures, but I'm going to focus on some of what we've been working most on, which include VOCs through our Superfund, and POPs through other grants, including that R35, and then along the way cover some key environmental health concepts that impact this work, including concepts related to endocrine disruption, obesogens, metabolic disruptors, and more recently some work from our lab that we call signaling disruption, and two-hit models that are relevant to that. And so briefly what I mean by these two-hit models, and really for liver is a tolerant organ. So I work with liver transplantation a lot. So you can take it out of the body, leave it on ice for 12 hours, put it in somebody, it does relatively well. You know, we probably all know people that drink way too much that never run into significant liver problems. So it's a tolerant organ, and sometimes it takes 2 insults to get liver disease. And so kind of one of the models we've been working under, in this case, volatile organics and POPs like PCBs. And this was from a review article which we wrote recently. A lot of the data we reviewed, some of it actually came out of the UK Superfund with Mike Petrello and Bernie that have been looking at liver endpoints. But we think these serve as a first hit by causing organelle dysfunction, abnormal function of various receptors, including nuclear receptors, which lead to a variety of responses that compromise the liver, making it very vulnerable to a second insult, such as a high-fat diet. And so we've got some evidence from at least animal models that some of these exposures don't really have any impact on liver histology at low levels unless they're combined with a bad diet. And so we think that while many pollutant levels in the environment are going down, and liver disease is going up, that it's the increase in the diet-induced obesity occurring on this compromised liver, which has been compromised by exposure. And that's why we may be seeing one reason why we may be seeing an increase in liver disease. So what's the historical perspective on this? And I got into this area about 10 years ago, and really at that time, if you picked up a toxicology textbook and looked up liver, you'd see stuff like this, these classic poisoning events. And so turkey X disease was aflatoxin. So I think many people in the room probably know about aflatoxin and liver cancer. What some of you may not know is it was first described in turkeys in England, turkey X disease. And a lot of this stuff happened in England. There was Epping jaundice, which was flour contaminated by methylene dianiline, which was transported in a van next to some flour. It spilled onto the flour. The flour was delivered to a bakery. Bread or whatnot was made out of it. People ate it and turned yellow. And then in Louisville, and this is really how I got started in this, was related to this event that happened in the 70s, the hepatic hemangiosarcomas, which are a very rare form of liver cancer that occurred at a polyvinyl chloride production facility, same one that Paul's brother worked at. And so here's an example. These cases still come in, 30, 40 years after the exposure. This is a CT from a patient I took care of that has this whole thing's, this liver's here, and this is the cancer. It's just huge liver cancer. And then there was a toxic oil syndrome in Spain where someone tried to turn the lipid in canola oil to modify it to become the lipid in olive oil, because olive oil, you could sell more than canola oil, costs more. And so I can't remember what the fatty acid is, but they were modifying the fatty acid to make a profit. And then there was some contamination in the oil, and that caused a fibrosing disease, kind of like scleroderma, but some people did get liver disease as a component. So this is the historical context, but my patients don't come to clinic. I'm a very active clinician. They don't come to clinic saying I ate contaminated flour where someone spilled stuff on it, or I ate bad olive oil. They come in saying they've got a belly full of fluid, bleeding varices, multi-organ failure. And so these are what patients actually present with. And so I'm trying to, under this river award, want to try to move the field away from these, very interesting but rare poisoning events into looking at more of the types of patients that physicians see in practice. But what we do know is that there's an epidemic of liver disease. And there was a recent publication a year or two ago showing that in the US, deaths from cirrhosis and liver cancer have increased by 65 and 50% respectively over the past decade and a half. And that's in the US, and the single worst state in America for increase was Kentucky. Why is that? I don't know. We've got a lot of reasons potentially, but perhaps environmental exposures play a role. I'm not suggesting it's the only factor by any means, but people don't even consider it as an etiology, and I want to change that. Just consider it. And so so what's our environmental health vision at U of L currently? And this really provides some of the context for the work that I'm doing. And so now we've integrated everything under an Envirome Institute, which is led by Aruni Bhatnagar. And so Envirome has a really comprehensive view of environmental health in terms of physical health, nutritional health. You can kind of read the spokes. And the little environmental health node of this real comprehensive model, we have several resources such as our Superfund. We've got a P30 that we'll, I guess, hear about in 2 weeks. That's led by Chris States. And I have this, I can't remember what RIVER stands for, Revolutionize, Innovate. I don't know what the rest stands for, but it's a neat grant. It's an 8-year grant. And it's actually no specific aims. So it's really fun writing a grant with no specific aims. But the cool thing is I tell people I'm the best collaborator because whatever your idea is, I can do it. I don't have to write a new grant to study that. And the other thing that's makes me a good collaborator is I'm locked out during the period of this grant from applying for a non-solicited R01 from NIEHS, meaning if we collaborate and you have an idea, you get to be PI if it leads to a grant. So I can apply as a PI for a solicited grant, an RFA, but for an unsolicited like R01 or R21, I can't. So we've really, just like UK, we've really increased our EHS focus. And kind of the approach we've taken with this river, we call it the environmental liver disease river, is we really do collaborate with people. And so we collaborate with agencies such as NIEHS and NTP. Made the slide a while back. I'm glad I had Bernie up on here at the top. I didn't go back and change it just because I was here, but many academic centers, societies, EPA, not as much with industry, but some on biomarkers specifically, and then CDC and ATSDR. So really trying to get people working together and going. Our approach is, because this is a new field, we do epidemiology studies to identify what relevant compounds may be through association studies, and then we go to the bench and develop animal models for them with the ultimate goal of bringing some knowledge and information that we can take to patients at the bedside. So what liver diseases do we study? There's lots of liver pathologies. The main thing we've been focusing on now is this toxic and associated fatty liver disease, or toxic and associated steatohepatitis. So this is a broad category of fatty liver diseases, initially recognized for alcohol. So everyone knows people drink to get liver disease. Alcohol puts fat in the liver. And then back in the 1980s, it was observed that some people got this pathology even if they didn't drink, and that was called non-alcoholic fatty liver disease. And at least in the US, most of those subjects are overweight, and many have diabetes. So that's believed to be, in general, the liver manifestation of the metabolic syndrome and obesity. It was later shown that certain chemotherapies and medications can cause it. They call that chemotherapy-associated steatohepatitis. And then I noticed that chemical workers from our Rubbertown area were getting this too. And so I propagated this series of poorly named diseases and coined this term toxic and associated steatohepatitis. So there's different, I guess, stages isn't the quite right word. It's really a pathologic spectrum from steatosis or fat accumulation to some will get superimposed inflammation. And that's what the itis means. Steatohepatitis is liver, fat, inflammation, steatosis, just fat. And then some will go on to get cirrhosis and some will get hepatocellular carcinoma. So the pathology is identical no matter what these causes are. So there's many roads that lead to this. And the fatty FLD, alcohol or toxicant, or the non-alcohol, this is really can reflect the whole spectrum, whereas the SH, steatohepatitis, is really, implies that there's superimposed inflammation. So this liver disease is always caused by an exposure. And at some point I'm going to get in an argument with NIAAA over this, but alcohol is just another exposure, just like diet, just like chemicals, in my mind. So what pollutants do we study? So this has been really fun. So we've done a lot of epi studies, again, trying to find out what chemicals to study. So we could pick a chemical. I like lead, so I want to study lead and see if that has effects on the liver. But we took another approach. We try to just do epi studies, not looking for anything in particular, and seeing if there's any associations with liver disease. And then, so we've been in the starting, we're using cohorts that already had previously measured exposures, because historically the exposure assessment was the hard part. So we started with exposure cohorts and then added liver biomarkers to it. In some cases they were already done and not analyzed, like ALTs. In other cases we had to measure new biomarkers. But these have been a lot of fun because they make movies about this stuff. And so these are examples of some of, not all of these are published, but we've done occupational cohorts, residential cohorts, NHANES, general US population, and we're now moving into what I call precision medicine and using clinical cohorts. And now we can add exposomics onto clinical cohorts. So, and Andrew's here, so ability to measure things and add that to clinical cohorts is really becoming possible now. And I'll show you a little bit of data, but anyway, this is a lot of fun. We've looked at a lot of things, and most of the time we find something. It's not always what we expect, but we oftentimes find things. So these are just association studies. They don't prove causality, but then we can select chemicals for further study. And so what are some of the chemicals that have been identified? Kind of as a leader in the field, I keep a list of this stuff, and this is just not everything, but you can see many common environmental contaminants of concern. I've listed the substance priority list rankings here. So many high-ranking chemicals have been associated with fatty liver in epidemiologic or laboratory studies, including many of the ones that you study here. And so, how did I get into it? So I think this is a neat story for, hopefully it's a neat story for trainees. I think it is. So I got started in this because I found this box one day and I opened it up and found slides and looked at them under a microscope. And so that's, been working on this new field, and this was purely by accident. I was a trainee. So what happened again was these angiosarcomas in Rubbertown in the mid-1970s. And so they started making rubber in Rubbertown in the 40s and World War II. So Japan cut off the Pacific shipping lane, so the US couldn't get natural rubber to make tires for military vehicles, so they started making synthetic rubber, and they made it in places like Louisville that were inland that couldn't get bombed by the Japanese or the Germans. And so, in some of these operations, vinyl chloride was used, and in 1974, liver cancers were first reported in JAMA in some of these workers. And then Carlo Tamburo was a hepatologist that was recruited to initiate a massive surveillance project in the chemical plant workers. And we've now had 26 cases in this one plant. There's probably more we don't know about the incidence of this tumor. Is about 20 cases per year worldwide. We now have 20 from this one plant in Louisville. So this is something that should be in the toxicology boards, should be in occupational health boards. This is a sentinel event in all of it, and really occupational medicine, because you don't get this cancer if you don't work at a plant. Interestingly, they've never lost a case. They've only been sued once. And now I know why, because I was called as a witness kind of during discovery phase, and had to meet with about 20 defense lawyers. Probably making 500, 1,000 bucks an hour just to not pay out one case where they clearly killed this guy. Very interesting. But anyway, what I noticed in the slide, I came across that my boss and long-term mentor, Craig McLean, had asked me to update the liver cancer experience in the cohort. And so I was snooping around and found this box of slides. So Carlo initiated medical monitoring surveillance. He was a pack rat. He saved, he's deceased now, but he saved over, I think 12,000 blood samples and other biologic materials from these workers that were getting annual draws. And so Craig and I kind of supervised that occupational biorepository. And I was snooping around trying to write a paper as a GI fellow on the cancers, found this box and looked at it. And what these workers actually had was classic steatohepatitis. And so that's when we kind of coined this term TASH. I'll show you some pictures of the slides in a minute. And so I thought I was really smart. And then later I found this letter here from Mount Sinai from Hans Popper. He was arguably the top human liver pathologist history of the world. In fact, when we go to the liver meeting every year, there's a Popper Memorial Lecture to talk about pathology. And Tamburo had sent some of the slides out of this box to Popper. And so this is what Popper said to him. I think it's in 1979. He says something like he's going to France and he's got to be brief, but he looked at a couple slides and the first showed fatty liver hepatitis. There was steatosis, necrosis, fibrosis. Looks like alcohol. If this is firmly excluded, I would consider toxic injury, and I hope to receive follow-up. And I don't think they did much on that, but I wasn't as smart as what I thought I was. This was noticed back in 1979. I was 5 years old at the time, so you didn't say when I graduated from medical school. So sometimes these famous old investigators, you got to give them credit. But to my credit, I did something about it and pursued it. This is what the biopsies look like. Fat droplets. Look at all this inflammatory infiltrate. This worker had a ring of fibrosis around the fat. He has cirrhosis. And these were non-drinkers. At least they reported not drinking. And the prevalence of the steatosis was 84%, and the steatohepatitis is 80%. And so to my knowledge, this is the highest. It's a small cohort. There's just 25 that I found slides on. But to my knowledge, this is the highest incidence of steatohepatitis ever reported still. This was reported about a decade ago. Steatosis, 84%. This is what you see in bariatric surgery patients. That's normal. But to have inflammation or have over half of them have fibrosis, this is incredibly unusual. And so we coined this term TASH to describe this. And about this time, or even maybe a few years later, there were some Brazilian authors that were reporting some similar findings from VTEX exposed workers at a plant in Brazil. So that was consistent. And very recently, there's been a epi study looking at fatty liver disease in children living near a petrochemical complex where they did a urine vinyl chloride exposure biomarker, and it was associated with increased odds for NAFLD, for pediatric NAFLD. So it's kind of good to see, you know, people starting to pick up on some of our earlier work and looking for this in children now and actually finding associations. So we've clearly started in occupational health. Here's vinyl chloride. It's got this chlorine group, which is removed. It undergoes metabolism through CYP2E1, just like ethanol. So this chloride's removed and it makes an aldehyde eventually, an epoxide, then an aldehyde. So we've been working on biomarkers, and this has been really a thorn in our side. So these workers that had TASH, their AST, these ones with these horrible liver biopsies, they had normal liver enzymes. And we've seen this repeatedly with exposures, is that these tests just don't seem to be very sensitive. And so along the way, we've had to work on biomarker development that we think, you know, biomarkers that we think will perform better. In this case, this necrosis biomarker works well. This is a cytokeratin 18. It's a cytoskeletal protein, which is a spillage protein. It's in epithelial cells, but it's enriched in liver, which is the biggest epithelial-derived organ in the body. And so when the hepatocytes die, they spill it. And if they die an apoptotic death, caspase-3 will cleave this protein, and there's a caspase-3 cleaved version called M30. And so if this is high, it's apoptosis, liver apoptosis. If this is not high and this is high, then it's necrosis, or at least not apoptosis. And so there was a lot of necrosis in these workers. The workers also had a lot of inflammation, systemic inflammation, as you would expect in their blood, with various cytokines being elevated, some insulin resistance, and altered adipokines. Like their adiponectin was really low versus controls 14, even though their BMI was only like 25. So there's probably some targeting of adipose tissue accounting for some of this liver disease. So kind of mechanisms for this that we're observing in humans using kind of these, you know, older but still somewhat mechanistic biomarkers was that the people that were exposed that had this disease had high levels of necrosis, pro-inflammatory cytokines, insulin resistance. There was antioxidant depletion, low adiponectin. And we repeated this in other cohorts of workers from other plants in Louisville, like these are ABS, acrylonitrile butadiene styrene workers, and similar findings. So we've been using more omics-based approaches. This was an earlier study that we did, just illustrating that, where we got blood from exposed workers. This was actually done by Metabolon a long time ago, vinyl chloride exposed workers. I had some money that I needed to blow through quickly from an expiring grant, and so Metabolon is really good about that. If you have money you need to spend in a hurry. But I'm not sure they give you the best data, but in terms of how many analytes you get. But anyway, we found some changes, and then doing a pathway analysis, we see that there's some, looks like there's some alterations in AMPK and AKT signaling. And so what our general approach is to make some initial observations using human data like this, and then try to look at it in animal models. So again, these are just associations. They don't prove causality, but at least in an animal, you can prove causality if you give an exposure that's controlled and see an effect. That doesn't mean it was causal in the humans, but that's kind of the approach we've taken. So a couple of these main hypotheses is metabolism disrupting chemical hypothesis. So this has really come out of the endocrine disrupting chemical hypothesis. So the endocrine disruptors change or impact hormone action, any aspect of it. But the metabolism disrupting chemical hypothesis extends that, and that chemicals have the ability to promote metabolic changes that can result in obesity, such as obesogens, type 2 diabetes, or fatty liver. So you can have potentially effects that are hormone independent that cause, or at least associated with, metabolic diseases. And so an example of this would be, say something targets mitochondria, mitochondrial toxin. You may get fatty liver, independent of any hormone action, because the liver can't oxidize lipids. So that would be an example of that. We've talked about the two-hit hypothesis, and sometimes the liver requiring two hits sometimes. Now clearly, if the vinyl chloride in the workers was causal, they didn't need a second hit because they weren't obese. But I failed to mention their exposures were astronomical. These guys were getting 1,000 ppm, and now OSHA limits this to well below 10 parts per million. And they were getting anesthetic doses. They would go in and clean reactors until they passed out. You can give vinyl chloride and do general surgery if you give it at high enough doses. And that's what they do. They'd go in, pass them out, then they'd pass out, then they'd pull them out and send someone else in. And they were otherwise young, healthy guys, because this was the worst job in the plant, and everyone knew it was the worst job. So once you had seniority, you didn't have to use it. So these guys should have been protected from healthy workers, because they were the new hires, the youngest and presumably the healthiest. But they were not. And then, so this part of the metabolic disruptor hypothesis is that a second hit, like a bad diet, may be required. So Juliana Bayer was a COBRA awardee for our liver COBRA, and worked with her to develop animal models of low-dose vinyl chloride related steatohepatitis to see if we can replicate any of these observations that we made in humans, and we could. So she gave a sub-OSHA level, 0.8 ppm, so it's much less than the 1,000 ppm that the workers were exposed to. We wanted to try to be more relevant to modern situation. And so this is an oil red O stain, lipid stains red. This is a low-fat diet, low-fat plus vinyl, high-fat plus vinyl. And so vinyl didn't do anything unless it was a high-fat, and then you can see more lipid accumulation. And she was able to block this by giving a ALDH2 activator, ALDA1. This will get rid of reactive aldehyde metabolites. And so we think it's probably the reactive aldehyde intermediate that's contributing to the toxicity, in much the way that acetaldehyde contributes to alcohol toxicity. This chloroacetaldehyde is much worse. That having that chlorine group makes it much more powerful. And so we saw insulin resistance in humans and diabetes. We can confirm that in the mice. These mice actually get apoptosis and not necrosis like the humans did, but that appears to be a dose-dependent effect. At lower VOC doses, even in human cohorts now, we're seeing apoptosis rather than necrosis. Inflammasome activation leading to high cytokines, oxidative stress. study here showed AMPK and AKT signaling alterations. We've shown that that was the case in mice, so just the exposures are altering metabolic set points. I'm sorry, I got a jumpy finger. And then there was a obesity interaction. A second hit was required, and we recently published on sex differences. So these are the plant in the 1970s was very sexist and only hired men, and mostly white men. And that was the plant hiring practices in the 70s, and that was a wrong choice in sex, at least if the men and women behave the same as mice, because the male mice were much more sensitive than the female mice to these effects of the exposures. So we've been working in our super fund on VOCs and also looking at liver, and we've recruited a VOC exposed cohort from Louisville, about 700 and 800 subjects. And while we were recruiting that, we wanted to have something to show. So we partnered with Dale Sandler at NIEHS and Larry Engel and Emily Werder, and looked at this Gulf cohort. So Gulf was a Deepwater Horizon disaster response cleanup worker cohort. So they measured BTEX, benzene, toluene, ethylene, xylene, styrene, and metals. And we did a pilot with them and got, I think it was about 150, 200 subjects, and went back and looked at, added some markers of liver injury, inflammation, endocrine function in a subset of these Gulf participants enrolled in a chemical biomonitoring survey. So the Gulf participants, there were many, many thousands of them, but not all of them had the blood levels of the pollutants measured. And so these were in the CBS study. And since it was a small study, we limited the number of confounders by just looking at males, non-smokers. You can see the rest of them there, but we did stratify across a range of toluene. And so in this, based on some of the animal models, so, one of our approach is to go from humans to animals, and then we learn something from the animals, and then we want to go back to the humans. So since we knew that these were going to be lower level exposures, and I'll get to that in a minute, but we wanted to look at obesity. So in our statistical analysis, we looked at obesity interactions in this cohort, and there ended up being 108 obese subjects, 106 non-obese. This is currently under review now. So here's the demographics. Most of them did work on the Deepwater Horizon oil spill, but at the time the blood was drawn for exposure assessment was about 2 to 3 years later after the exposure. And so they've gone back to show that the levels of the BTEXs were really similar to NHANES. And so they really, these things would have been metabolized. They were presumably high after the exposure, but 2 or 3 years later, they're gone, at least the BTEXs. So this is similar to NHANES. And so what we found was that of all the BTEXs, only benzene was associated with this liver biomarker. This is the apoptosis one. So remember, the high-level vinyl workers got necrosis, but the low-level mouse got apoptosis, but only with a high-fat diet. And so this was similar with benzene. It had increased liver apoptosis. These are adjusted beta coefficients, but only in obese workers, not in the overall group or with the non-obese participants. And there was a significant interaction with obesity and exposure. And that increased liver apoptosis was associated with increased inflammatory biomarkers, IL-1, 6, and 8, but again, only with obesity. And we haven't done a lot with metals, but we always find stuff with metals. I mean, they always come up, so metals always positive. Anytime we look at them, we always find something with metals. So they were similar. All these biomarkers for lead and cadmium disease biomarkers were increased, some of them even in the overall cohort, but again, especially in the obese ones. So again, we're going from a chemical plant observation, going to an animal model, learning some things, and then applying what we learned to other cohorts. So it was really interesting to me anyway, or not interesting, but felt good that somebody's picked up on this. So we actually made a medical guideline, which I don't think we're ready for that kind of prime time yet, but the European Association for Study of Liver Disease just published occupational liver disease guidelines. And so they talked about the taffold and the angiosarcomas and vinyl chloride, and cited our work. which we've come a long way in 10 years from finding that old box and wondering if we were just imagining things or was there really something there. So that was rewarding. We've gotten into POPs, and so we did this study. This was actually a P30 funded study. So this was U of L had a P30 with Ken Ramos, and so I was a career development awardee. And so I wanted to get out of the chemical plants and into general population, and not being an epidemiologist, I needed help, but I knew NHANES had measured pollutants and they measured ALTs, and I wanted to look at associations. Others had done this for diabetes and lipids, but no one had ever done it for liver. And so we did this study looking at associations between exposures and unexplained ALT elevation. So these are people that have an abnormally high ALT, but they don't have a reason to have an abnormally high ALT, like they're not, don't have Hep C, don't have Hep B, don't have hemochromatosis. Those are all measured in NHANES. They're not excessive alcohol drinkers. You exclude all of them. And so this has been suggested to be a biomarker for fatty liver. Jeannie Clark had published on that. And so we're looking at things specifically that had dose-dependent increases. And so these are odds ratios for unexplained ALTs. We've used, it's actually very controversial as to what constitutes a abnormal ALT. So we used a couple of different cutoffs. So one was published about 10 years ago in EHP, and then we used another cutoff that Van Rita Wong, who was a postdoc in Bernie's lab, just published in the last year. But we found PCBs were associated with increasing odds ratio by exposure quartile, both the non-dioxin-like and the dioxin-like ones, and then some organochlorine insecticides, and again, some metals. I mean, we always find metals there. And so when I first got these results, I'd asked what in the world a PCB was, and Bernie was very helpful and referred me to Larry Robertson, who was very helpful. And but this was encouraging. This was actually, a lot of people do PCB research here at UK. I don't know if any of you ever looked at the MSDS sheet from Monsanto on PCB, but this is what it says. Consistent finding in animal studies is that PCBs produce liver injury by prolonged and repeated exposure by any route. Liver injury is produced first and by exposures that are less than those reported to cause cancer. Thereby, exposure to all routes should be kept sufficiently low to prevent liver injury. So I was confident we were onto something, because if Monsanto says something has a bad health effect, it probably does have a bad health effect. So we started looking, and this was the first study. Here's a picture of Van Rita when she was at UK, and she had done this study before she came over to UK, and we worked with NTP on this. But similar to that vinyl study, we did control diets, high-fat diets, with or without, this case, PCB 153. I didn't know about PCBs, or I might have looked at a dioxin, but I just figured we'll do the 153 because that's the highest level of all the PCBs in humans. And this one was an obesogen, so you can see the visceral fat, but it's an obesogen only on a high-fat diet. On a control diet, there weren't any obesity modifying effects. And so we showed that there was a diet interaction, there was necrosis, there was some endocrine disruption with altered insulin and leptin, there were alterations in cytokines, and she recently published it's worse in females. So it's the opposite of the VOC, which was worse in males. So we've been working on aniston studies. For sake of time, I'm going to be quick and just flip through some data. So aniston studies measured PCB congeners. This one's published, and so we went back and measured, since we found increased liver necrosis and fatty liver in the mice fed high-fat diet and exposed to PCBs. We looked at associations between 35 non-dioxin-like PCBs and the necrosis liver biomarker M65, and we found associations for multiple congeners, multiple positive associations. And we chose to use the aniston cohort because the BMI in this cohort was high. 80% were either overweight or obesity. So they're kind of like unfortunately, the subjects were kind of similar to the animal model, and that many of them were overweight and obese and had a high PCB exposure level. And so those with the necrotic liver disease had insulin resistance and pro-inflammatory cytokine elevation, and also alterations in insulin and leptin. And so the human data were pretty concordant with the data that we developed in the animal models. showing that there was an effect on endocrine disruption and liver disease and altered metabolism and inflammation. One of the criticisms from these types of studies is there's no histology. Histology is the gold standard for liver studies, just like the chemical workers that had biopsies. These types of studies, when you add liver endpoints retrospectively, you can't go back and add a liver biopsy, and you can't put a liver biopsy in a residential cohort study of unselected people because the IRB would never approve it, and if they did, you'd get shot if you tried to do it. But so we've been looking at creative ways to get around this, and so we worked with Brian Chorley from EPA using a panel of high throughput liver specific microRNA biomarkers. Well, not always liver specific, but microRNAs that have been associated with liver disease. And so the concept is you do a panel, you get some that are abnormal, and so we have some that are abnormal here, some up, some down, and then you plug them in IPA, and the IPAs now have a module where it'll predict pathology. And so the pathology showed that the ones that we characterized as having TASH based on these cytokine and CK18 panels, that the IPA, the microRNA changes associated with those groupings predicted steatosis, liver cancer, low albumin, cancer, inflammation. So consistent with fatty liver and steatohepatitis. So we're working with Andrew now and Bernie with Aniston cohorts, and so we've now gone back. This was a pilot study. We've now gone back and measured microRNAs there, and we hope to work with them to look at PFAS associations too. Yes.
Is that a deep sequencing where the microRNA
so this,
is coming from the serum? But
yeah, so let's talk later on that. It's a different technique. We can talk. I'm going to run low on time, so let's talk after if you got a minute. So Josiah Hardesty's here has been working on using proteomics. He's a very bright UK graduate that got a PhD in biochemistry in my lab, and he did proteomics and PCB treated mice. And so, if you do proteomics, you can do some kind of transcription factor analysis, just like you can with RNAs, to try to predict what transcription factor changes would have produced the changes that you see in the protein levels that are measured in the liver. And so he had this data for a long time and couldn't figure it out. And so this is one reason why you want to work with a physician sometimes that are engaged in clinical trials, because these transcription factors were all downregulated. And so these are the transcription factors that are targeted in all the phase 3 and 2 NASH clinical trials. So it's not like there's one mechanism, it's like all the mechanisms that are bad are being targeted. So the medicines are agonists to make this stuff go up, but the PCBs make it all go down. And so this is an example of ways that the liver is compromised. These are all protective responses, anti-fibrotic responses, antioxidant responses. They're all down-regulated by the exposure. And then when you hit it with a high-fat diet, they're more prone to get a more advanced fatty liver. And how could that be possible? Just briefly, had done some phosphoproteomics. And just like you can do a transcription factor analysis on the raw data, you can do a kinase activity analysis. And so all these kinases, high-fat diet doesn't do much. The exposure downregulates kinase activity. And if you have the exposure on a high-fat diet, you have a massive downregulation in the liver of kinase activity. And so that's what we call signaling disruption. And many of these transcription factors have are phosphorylated. They have phosphorylation sites. And so how could this be possible? So Josiah went on to show in his dissertation that there's predicted binding sites using docking software for various PCBs on the epidermal growth factor receptor. And he developed a screening assay for EGFR inhibition, which we think of as a cancer receptor, but it's very homologous to the insulin receptor, and it has a lot of metabolic activities and properties in the liver. And the PCBs are incredibly potent antagonists to that in the screening assays. So you can kind of do kinetics, and they have femtomolar potency actually in inhibiting EGFR signaling. And last paper from this graduate work that we got to get out is he's able to attenuate the fatty liver by administering exogenous EGF as a therapy for PCB liver disease. I'm not suggesting that's a therapy, but it just helps to prove the concept that PCBs contribute some of their liver disease probably by antagonizing the EGFR, leading to inhibition of the phosphoprotein signaling all the way downstream to nuclear receptors. Not quite proven yet, but supports the concept. And so he's coined this term signaling disrupting chemicals, looking at phosphoprotein signaling. You can kind of see some of these signaling cascades here in homology between insulin receptor and EGFR. Let me just cover one last thing, and I'll take some questions. We've been working here with dioxins, and I'm going to skip over that. And aniston too, and we find things with dioxins too. I can come back to it if people have questions afterwards, but I want to wrap up. I think this is important. Hopefully it's important because I've got a grant on this out with these other investigators. What do they call it? Victor grant. That's under review. So we're using these exposure cohorts to look at liver disease, and historically, again, we were doing it because it's harder to do the exposure assessment than the liver assessment with some of the serologic biomarkers, again, which are limited because they're not biopsies. But again, my patients don't come to me saying I'm from Anniston, Alabama, or I've got a paper from the C8 cohort. I'm from Parkersburg, West Virginia. Maybe they come to UK or Parkersburg, I guess, and then we come to UK. But and if you ask them if they have exposures, they just look at you like you're crazy. And so what I wanted to do was look at exposures in unselected NAFLD patients that come to clinic. And there's not really many studies looking at this, although Marion Voss and Dean Jones just published one with PFHXS in human children associated with worse NASH in pediatric patients. And so worked with Naga Chalasani, who's the lead investigator in the NASH Clinical Research Network, who had to do a pilot study, and he had some well-characterized patients from his NASH clinic. I could recruit some, but he had a bunch already ready, and shipped them to Dean Jones to do metabolomics and exposomics on him. And I'll show you some of the preliminary data. Exposomics is just exposome or omic scale characterization of things including pollutants. He reckons he can get, I think, maybe a couple, at least a couple hundred different environmental exposures with a drop of blood. They do LCMS and GCMS. I'm going to show you some LCMS data. So anyway, the hypothesis was that NAFLD is a multifactorial disease associated with abnormal hepatic metabolism, and exposures may influence NAFLD severity. So we're not asking, does the patients already come to clinics? That's what I see. So we're not asking, does the exposure cause the liver? These are people that are in clinic, and they're in my clinic, or in Naga's clinic in this case, because they already have the disease. But there's a spectrum of disease. So is it bad if you have exposure, or is it, or some exposure protective? And so that's what we're looking at. So this was a cross-sectional exposome-wide association study, basically, or a metabolome-wide association study in 150 adult NAFLD patients with very well characterized. And they were characterized, in this case, by imaging. So many of them had biopsy, but all of them had FibroScan, which is a imaging biomarker that is widely used around the world. That gives a readout on steatosis called controlled attenuation parameter, or CAP, the amount of fat in the liver. But what's most important is the amount of fibrosis. So it's clearly been shown multiple times that fibrosis is what drives death and fatty liver disease. And it's not just liver-related mortality, it's also strongly associated with cardiovascular events, not just having the disease, but having fibrosis in particular. And so the main these were the two endpoints. So the EWAS and MWAS versus steatosis and fibrosis, with fibrosis being the most important. This is what the FibroScan looked like. It's a probe. They have a portable unit too. I was glad to see that Indiana patients were even bigger than some of my Kentucky patients. So we're from Indianapolis. BMI 36, 51-year-old, mostly women. 51% diabetic, very sick cohort. The liver stiffness measurement was 12. That correlates with stage 3 fibrosis. Stage 4 is cirrhosis. So very advanced fibrosis, and this is in severe steatosis range on the mean. So these are very sick liver disease patients. And so this is what, say, the MWAS looks like. So there's a clustering heat map. This is MWAS versus this one's liver stiffness measurement. So this is fibrosis. This is the maximum scores around 75. So you can kind of see some patterns emerging. And these are, this is just the LCMS data. They measured 31,000 features, and about 130 were significant at a FDR 0.2. About half of them were upregulated, which were in blue, and half were downregulated. And then we do this is versus the liver stiffness measurement or fibrosis, something similar here versus CAP or steatosis. And you can see some clustering there too. We picked up a little bit more significant associations here. 508 features were significantly changed, about equal numbers up and down regulated again. And so some of these were exposures, some of these features, some were just regular metabolites. You could pull the metabolites out and do look at pathway analysis to see which pathways are affected. And this isn't novel. This has been done a million times for NASH already, but I don't think it's been done for FibroScan. It's been done for biopsy though, which is better, but we get consistent results with the other studies, and that bile acid pathways and carnitine and beta oxidation are impacted with both of these. And the ones that were common for liver stiffness and CAP were the porphyrin. So all the porphyrin is just bilirubin. So if you have increasing fibrosis, your bilirubin goes up. And so you got to take this with a grain of salt, and I'm going to talk about exposure association. So you got to remember reverse causality. Clearly high bilirubin doesn't cause advanced fibrosis. Advanced fibrosis makes the bilirubin high, and that's why cirrhotic patients are jaundiced. But anyway, these are consistent with literature more or less that are consistent. But these are the exposures that were significant. The pluses, they're all positive associations. So the pluses are on the liver stiffness measurement or the CAP. So they're carbamate insecticides, azole antifungals. So these are like, diflucan that people take for yeast infection. They actually add this stuff to our food as a fungicide. You can measure it in people's blood. Here's some other insecticide that I don't even know what it is. There's a dibenzofuran. I didn't go over our dioxin data, but similar AHR agonist associated with liver fibrosis. This one, they've got to get a standard. So the authentic standards were only available so far for these two, nicotine and methomyl. They've got to get one for this. This is Pataday eye drops. So I have trouble believing that a eye drop is going to be associated with liver fibrosis, but nonetheless, that's what the data are. So we're in the process of validating this. And then the other thing that we're working on currently is to go back for these, and we can do an MWAS. We're doing MWAS versus these exposures. So we know what pathways are associated with increasing liver stiffness. We know what exposures are associated with increasing liver stiffness. And so if those So if the exposures change the same pathways that are associated with the mechanism, then maybe these exposures are acting through that mechanism, like through altering bile acid metabolism to increase fibrosis. But again, these are just association studies. So we're going to take it a step further in the proposed. We'll see how it reviews. I think it should review great. To take this a step further in the vector application. This is, I think, an approach that you can consider in any disease that you do. So it's, because I'm working on all these clinical trials for new drugs, I'm very much aware of this, and we recruited Nago specifically from IU specifically for this purpose, because he's got access to large randomized double-blind placebo-controlled studies. And so we want to do multiple time point, multiple liver biopsies, and multiple exposome and metabolome assessments. So these are great because what I propose is you take placebo-treated patients, and it's a natural history study. So does an exposure at baseline or changing over time lead to progressive fibrosis? So these, I think you can get more at causality because it's longitudinal. And in we know that there's no drug approved for NASH. The first drug will be approved probably this year, FXR agonist. It works 2 times better than placebo, and the response rate is only 30%. So most people don't respond, and is that because an environmental agent is interfering with its mechanism, which is FXR agonist, which I've already shown in an earlier slide was one of the receptors that PCB seem to target indirectly. And so we can do that in the active drug treated arm. And so we'll see how it reviews, but we're gonna have, I think, 2,000 subjects that we propose to do this on. And again, these are just the LC data. And there's many more exposures in the GC data, which we haven't analyzed yet. So I'll finish up with, close out on the case presentation of Paul Coder, my patient who has cirrhosis that worked at Camp Lejeune and had VOC exposures in his drinking water when he was stationed there, and had a brother that worked at Rubbertown and vinyl chloride and died of cirrhosis and liver cancer. He wanted to know if his liver disease could be related to the cirrhosis. It was originally attributed to his alcoholism, or not alcoholism, alcohol drink. He was a Marine. He wasn't an alcoholic, but he drank like a Marine back in the day. Said you drink a fifth, get up at 5 a.m. and run 20 miles. That was in the past, but now he's a little overweight and diabetic. That's what his liver disease was blamed on by people, including me. But you know, it's important to note that there's federal legislation on this fatty liver due to environmental chemicals. It's Camp Lejeune. What's the name of the? I don't know if the name of the act is on there, but there's certain medical conditions that if you lived at, you or your family suffered from any of these medical conditions, and you lived on base during specific years between the 1950s and 1980s for a specific time period, then you're basically service connected for those things. Which is, you know, I work at the VA, maybe others here too. You know, I've never seen a family member service connected for anything, but family members are actually service connected for some of this stuff. And so hepatic steatosis is on the list, and they can actually even sue for financial compensation, and that office is in Louisville, by the way. So I told Paul he should sue for financial compensation, and they turned him down. And I wrote a letter of support for him, and they cited all my papers, and there was a Institute of Medicine article on this that cited all my papers, and I pointed that out, and they turned him down anyway. So poor Paul, but he's doing well. So anyway, I want to thank a lot of people, especially my family. There's my son that I started with playing football here at UK, my wife who just graduated for college and nursing. Our lab is neat in that it's in this, you get a great view of Thunder over Louisville. And so there we are at that last year. I grew up with Andy Bashir. I was best friends with Andy's brother, Jeff. And this was taken at his inauguration day. We gave Andy hell. If I'd known he was going to become governor, he was the little kid brother. I would have treated him better. My middle son, Sam, he likes to jump off of tall things. And my stepson, Nikolai, and my sister, who's actually an environmental lawyer at Stites and Harbison that likes to ride horses. And many collaborators and mentors that have taught me environmental health in red, and some people in blue that have been at least associated with UK that you know, am or have been working with. So a lot of UK ties. So I'll stop there. I know it's getting late, but I can take questions.
Great. Next question.
Yes.
Going back to that difference between necrosis and apoptosis, do you get an impression of, is it a time-dependent phenomenon or related to acuteness of exposure?
Yeah, so all I can tell you on the time dependence is, you know, so these are the biomarkers, specific antibodies that we use for the human studies don't pick up mouse very well, at least the apoptosis antibody. But what they do work well in is human hepatocyte cell culture. And if you add vinyl chloride metabolites to the cell culture, you see the total cell death biomarker rises up pretty quickly, although those concentrations may not, may be very high. A lot of these studies are chronic, and so, with the dietary studies in NASH, most people, so it's a little interesting working in this field because most tox studies always have short-term dose escalation studies, but most NASH studies are 12-week studies or 24-week dietary studies. And so that was kind of the approach that we've taken, is a little bit longer studies with lower dose. And so we have really insufficient time course data, I guess, to answer your question.
So obviously there's a ton of different environmental contaminants, and then when you add diet into the mix too, I guess what I'm wondering is, which ones do you think are most important? You mentioned metals popping up over and over again, but I think it's combinations of all the things, and you just have to pick one to study the mechanism, or I guess you kind of alluded to it with the drug treatment where, are you blocking beneficial effects from an exposure?
Yeah,
so like not really.
So I think the answer is going to be doing these precision medicine studies in people that come to the clinic that are tightly controlled and have extremely well characterized disease. So you can do that no matter what you study. You could use that approach. So you're a cardiologist and looking at, triple vessel coronary disease. I'm sure there's a clinical trial for that. That's longitudinal, that you can do exposome before and after, with or without drug study. I think those are going to be most important. And Andrew and I were talking before, just for Aniston cohort, for example. I mean, my complaint is that it'll never end, because now you've got PFAS, you've got dioxins, you've got insecticides. How do you design a multi-pollutant exposure model, which is the reality? It's probably all the different things and all the different diets. I mean, it's job security, I guess. Yeah, Andrew?
I mean, the stuff you talked about at the end, piggybacking environmental measurements onto these interventional clinical studies is hugely powerful, right? But
I hope the reviewers think so.
Well,
I do.
I don't think it's enough of it. But one of the reasons when we try to approach people about this is, of course, that they're concerned about whether the subjects are really consented for all of the environmental aspects. So I guess my question is, these studies we're talking about, is that part of the, you get some sort of broad thing that says we're going to be able to measure other risk determinants?
Yeah, so the study that we proposed to start with was the study from NIH funded study of so this company got a great deal. So Intercept, they'll be the first drug approved, but their first big clinical study was actually funded by the NIH, and they benefited from it. And so they clearly had a consent for a biorepository sample for future unspecified research. And many of the industry sponsored studies in this field now have that, because there's, again, no good blood biomarkers just in general. It's especially worse for environmental health. And so they leave these things in case, because they want, again, 1/3 of people the drug works on, you need a biomarker for futility or for stopping. And so they leave these things. So it depends on the study.
Because I think the FXR thing, it comes from some sort of genetic. I thought the way that it was discovered was from some sort of heritable genetic alignment.
Yeah, so
that's a whole other area of genetic modifiers in this.
Yeah, and in fact, you all had a couple of years ago, not necessarily you personally, but Mangelsdorf was here at UK, who just cloned FXR. I can't remember who invited him. Mangelsdorf and Clore from Southwestern.
I want to thank Matt for coming, and very interesting. If you want to stick around and talk to Matt, it would be fine.
Thank you.